frustrating. Anyway.
Big observation. That's from a previous application.
Oh no, this is neural formation.
Yeah, I think it's
It's just if you. Oh, that's animal bio, isn't it?
Gen bio, yeah.
Yeah, gen bio.
Yeah, but they like incorporate more of it.
Wow.
Like, it makes more sense. Yeah, like, plant bio, like, they do plant stuff, but it's more like stuff.
Just cell focus.
Yeah, just like a lot of basic stuff.
Oh, it's just going.
Into just plants. Yeah. I'll leave it. I feel like I didn't really learn anything from either of those classes.
Did you see your session? Yeah. Okay.
And then I stopped. Yeah, yeah, that'll be up by 5. I remember that. Remember that, John.
That's great. Yeah, the carbon. And like the structure is different. My mom was like, his structure of lecture is different from structure of textbook. Was like, no, let's throw the big structure.
Start it, but there's something. Unless he did it on a different document.
book is highly different.
I will.
I read it. I was like, huh?
I was just gonna let him do it because he was like, oh, I didn't I'll do it.
There's like a couple things
I didn't do it.
in the book, but like,
Yeah. I couldn't even, no. I was trying to download the software to do that and I was just not able, like, I'm not tech enough. I couldn't even get the thing downloaded on my computer. I just, yeah.
I was like trying to copy it, and it wouldn't copy it.
Oh, it probably wasn't compatible with the document or something. Yeah, I was like, I'm done messing with this. I'm
there's a
like,
protein called serapin that is responsible for cytosis and like
I know human genetics is directly.
responsible for us protein that is responsible for releasing neurotransmitters and all kind of stuff. But the thing is, we know they are activated by calcium ion, but we don't know exactly how. Like, so there's membrane, there's like a lipid, like
we come back from spring break.
so like there's it's like what that's a word. That's like what contains neurotransmitter. There's membrane, like a diffusion.
No, there's 2.
Open it up, and then like a
Yeah, those are the classes.
exactly how this opening happens or like stuff, and there's a protein that is responsible for like a pinching.
I have an example. Let's do that.
Like there are a couple of reviews on it or like a theories on it, and then like even Dr. was kind of interested in it, so like why not like just summarize or like did a review of like theoretical models, and isn't it fun? And we're kind of trying.
Yeah. Or just put more
But sometimes it's kind of difficult as shit. This whole chemistry and the protein structure, how they interact with the calcium ion, and like change the physical structure. Like, can you just diagram this? Like, I really have to translate it.
Yeah, like, wait, what is that? Where did we get? I honestly don't know what I'm doing at all. Oh.
Topic.
And then it's
What's your
so it's
topic?
like business and then
Yeah,
I don't
yeah.
know, and like I don't know. It's really just like
My family just, we don't wait. I'm gonna get and then we go, you don't want to.
Tomorrow is like, check it out.
I'm like I'm just worried about it today.
They don't have any other. At least you could pull out like 2 or 3 papers and bring it up.
I didn't get to do laundry yesterday.
Yeah.
I was like in the library all day.
Then what we do when it's in
Oh, yeah, I remember. Do you know all of them? I think that happening now. Like, what do you think?
I'm just gonna like
I was so relaxed. Oh my gosh.
Like Sunday night, not spending for reading for like 3 or 4 hours.
Yes. I'm so stressed.
Oh, Chicago, okay.
So
it's so bad.
So you can into the best.
Close down. that, but like they were like scattered all around. And you write down the whole thing and I'd be like, I already wrote that down. That's probably going to the A4 now.
Because I know I mess up this time, or like a couple questions, I'm sure I'm wrong. I don't know, that was kind of challenging exam. Easier was like, I didn't expect the average to be 77. That's kind of higher than.
Take the yes or no.
Maybe like a.
Yes or no. Just give me the yes or no.
Present. Like is good for you.
Yeah, but
There are 3 words.
Right.
3 words.
And one of them should be and.
there's so many rings. I was not prepared for it.
Yeah, that's so true. That's so true.
Vicky gave a lot of extra.
You wrote less than 4.
Are you going to?
Well, I'm either gonna let Ashton Bravo off, or I'm gonna let Jordan off. I'm not doing football anymore. I've decided that it's not good for my body. My ankles and knees are not.
I have to speak about it.
Well, yeah.
I mean,
Well, I see the model, but.
I like playing football.
I've heard.
No, I can do this later. And I have to try to keep up.
Shins are bugging. I'm literally gonna say that. Shins are bugging and lots and lots.
All right, let's go ahead and get started. How's everybody's morning going? Not bad? Surviving? Anyone do anything fun this weekend? No?
I don't have to be scared for exam.
That's good. That's always fun, I guess. In its own way. All right, cool. So today we are going to be continuing on in chapter 8. We're going to be talking about how, we'll do a little review on making proteins and then some protein processing in the cell. So how proteins are modified, folded, and degraded. And so the suggested readings in chapter 8 are sections 4 and 5 here. And so that's what we'll be going through today. Quick overview. Last week in lecture 7, we went through going from DNA to RNA, and we talked about RNA polymerases and how there are these sigma factors that help it recognize where to bind on the DNA transcript. And we talked a little bit about the genetic code and ribosomes and what they do, and then tRNAs, the transfer tRNAs that transfer the amino acids to ribosomes to make the polypeptide sequence. And then we went over translation. Today we're going to kind of go through another quick brief overview of translation, going from the mRNA transcript to protein, and then we'll talk about how proteins are modified and folded and degraded and how they are exported to certain areas in the cell. So I guess I can stop here and ask, is there any questions on anything from last week? Anything that you guys need me to go through again? All right. So it's helpful for us to look at what we've learned so far, and here we're taking a DNA template. So this is the DNA template, the double strand, and so here we're using an RNA. Can you guys see my cursor? No, I always forget that you guys can't see it. But yeah, so here we have the DNA template. We have a sense strand and the template strand, and so we're using an RNA polymerase here in order to make an mRNA transcript. So this is the mRNA transcript that we get. And so this is a 2-gene operon, which just means that there are 2 genes on this transcript. So once we get an mRNA transcript, we're going to have 2 proteins that are produced. And so this template strand is what is read by the the RNA polymerase. to make the mRNA transcript. So in theory, it's the sequence of this sense strand that will match the sequence of the mRNA strand. And so there are DNA elements that you guys should be familiar with. So there's the promoter sequences and the terminator sequence here. You don't have to know the -35, -10 in this class, but essentially these are some of the elements that you should know. And the promoter is important. You should know that this is where the sigma factor will bind. And the sigma factor basically lets the RNA polymerase know where to start transcription. So from this single transcript that's produced, we have two different proteins from the operon, so gene A and B, that are both driven by this one single promoter. And then, so if we go down to this mRNA transcript here, we have this untranslated leader sequence, and there's also a trailer sequence here as well, trailing at the end, that's also untranslated. So basically, this is just saying that in between this is the protein coding region. And so, yeah, so these untranslated regions can just help regulate, inhibit or promote expression, but that's not, you have to know all the details about that. But in the end, what's important to know is that we get these two proteins that come from the single mRNA transcript. And just kind of quick overview. And then we talked a little bit about the 3 stages of protein synthesis. We kind of talked about how we have initiation, elongation, and termination. So, you know, this is just getting back to the ribosome and its mechanism of translation. We talked about the ribosome having 2 ribosomal subunits, the 30S and the 50S, that have to come together to make the 70S full ribosome. And so that together, initiation, helps to place the first amino acid in its position. And then once that ribosome gets properly positioned, we have peptide synthesis that can start to occur and begin. And so just as a reminder, the ribosome is usually placed on the first amino acid position, and that's usually a start codon. And remember that start codon is the AUG or methionine that we talked about in the genetic code. So that's always going to be the start codon. And then you have this elongation, the sequential addition of amino acids, and then termination just finishes the polypeptide sequence, so it helps to release the completed protein. And so the ribosomal subunits can then be recycled for further use of protein synthesis. And so each one of these phases requires a number of protein factors and energy in the form of GPT. We're going to kind of touch on this in a little bit, but not too much detail. But you should know these 3 stages of protein synthesis and not confuse them with the 3 stages of transcription, because we have similar like initiation and elongation and that thing. So that's why I kind of want to reiterate this for you guys. And then we're just going to go through each one of these into a little bit more detail one more time, just to hone in on these mechanisms. So once again, in initiation, you have that tRNA molecule, the transfer RNA, and that is that adapter molecule that essentially will bring the amino acids to the ribosome to build that polypeptide sequence. But in order for this to start even working, it needs to be charged. And the way that it's charged is it is bound to this N-formal methional tRNA. So this is the methionine, the start codon, the first amino acid. So you'll see it come in variations, varying terminologies. You might see it as N-formal methionine or methional tRNA, or MET, MET, or AUG is the codon sequence. But essentially, this tRNA that's charged can then bind, and it's the only amino acyl tRNA that directly binds to the P site of the ribosome. And so once that happens, you should know these 3 steps in the elongation step of translation. And so these kind of just happen over and over and over again. They're repeated in a cycle. And the first thing is the amino acyl tRNA binds the acceptor site. A peptide bond is formed between the new amino acid and the growing peptide that's pre-positioned in the P site, which happened in initiation. And then once you have this peptide bond form, you're getting this movement down the transcript, and so the message will shift by one codon in order to have the uncharged tRNA now move to the exit site, the E site in the ribosome, which is ejected, and then it allows for another tRNA to come in through the A site. So it's just the sequential order. And from last lecture, if I had some images that would make more sense for you guys. And lastly, we have termination. So finally, the ribosome arrives at the end of the coding region, which is the stop codon. Remember, in the genetic code, there were 3 stop codons. You don't need to have those memorized. You just need to know the start codon. But there are 3 different ones, and they vary just by one amino or one peptide. And so after forming that last peptide bond, you have this release factor that binds and allows the completed protein to be released, and then the ribosomal subunits, the 30 and the 50S, can then be taken apart and recycled in the cytoplasm. Any questions on translation and all these steps? I have a lot of memorization there, but these are some of the things you should know. Remember the elongation, these are just repeated over and over again. Okay, so I do like emphasizing how antibiotics work since my dissertation work is on antibiotics. And so I work on antibiotics that don't target protein synthesis. I worked on ones that, like amoxicillin, I've talked about in the past. It actually targets the cell wall synthesis and the peptidoglycan layers in the cell wall bacteria. But I did previously mention that there are different antibiotics that can target different parts of the bacterial cell forming. And so a lot of different antibiotics target protein synthesis. And so there are different parts of the protein synthesis mechanism that different antibiotics can target. So I'll kind of go through a couple of these. You should know the mechanism in very brief understanding. You don't need to know the structures at all of these. So if this is streptomycin, for instance, and so one thing I want to definitely emphasize is that given that the importance of protein synthesis, and given that the ribosome in bacteria is very different from the ribosomes in our cells and eukaryotes, the ribosome and this protein synthesis process is an excellent target for antibiotics because it's a fundamental process that the bacteria needs to make in order to survive. And by targeting those aspects of the bacterial cell, there's little chance of crosstalk with the eukaryotic ribosomes. So we're not targeting our own ribosomes, our own machinery for protein synthesis. We're only targeting the bacteria, and that's really important in antibiotics. We have to exploit mechanisms that we don't also have. So here I'm showing you the structure of streptomycin. Once again, you don't have to have this memorized, but the way that streptomycin works is that it's able to bind to that A site. And so when it binds to the A site of the ribosome, it basically allows for improper the codon-anticodon matchup, and so that results in the wrong protein sequence. So basically here, translation still will be able to occur, but it will eventually kill the cell because it's resulting in the wrong protein sequence. So it kind of screws up with the ability for the tRNA to bind to that codon-anticodon alignment. So it binds in the site where the tRNA should. So that's streptomycin. Tetracycline is similar to streptomycin. It also prevents the charged tRNA from binding to the A site, but instead of binding right directly on the A site, it just binds near it, and it just prevents the charged tRNA from binding entirely. So it acts as a steric hindrance by blocking the A site. Whereas in the streptomycin, it can bind the A site, but then the tRNA can bind and then it ends up translating the wrong protein, whereas here it's completely preventing the charged tRNA to bind to the A site because it's blocking it essentially. So for, memorization purposes, you should know, does it bind near the A site? Does it bind the A site itself? And then do you get a resulting protein sequence? Is it the wrong one? Is it the right one? That kind of thing. And third, we're here, I'm showing you chloramphenicol. It's just another antibiotic, and this one's a strong one. So basically this binds to the P site and inhibits the peptide bond formation. So it binds to the second step of where the tRNA is binding in the ribosome, the P site. So the tRNA can still get into the ribosome, it can bind the A site, but then when it's getting to the P site and it's trying to give the ribosome these amino acids to be able to build that peptide, it's preventing the enzymatic activity of the ribosome, because in that P site is where you have that activity. So you're not getting a peptide sequence. And lastly, I have erythromycin. And so this one, if you can see here, it has this like really massive ring structure of carbon atoms. And so this drug binds the P site as well, which here I call the peptidyl transferase site, just so you guys can get used to different terminology. But basically, by doing so, this one also alters the peptide bond formation, and it screws with the amino acid sequence that's coming out of the cells. So, or out of the ribosome, sorry. So these two both bind to the P site. These ones bind either near or on the A site, and so here you get mistranslation. Here you're not getting the translated protein. And these will bind to the P site, so this one will inhibit the peptide bond formation. We're not going to go into the mechanisms of how this happens, but this one, erythromycin, will also bind the P site, but it doesn't inhibit formation, it just alters the peptide bond formation. So all of these different mechanisms are slightly different, and they have different targets within the ribosome, but ultimately what's happening is it's ultimately destroying protein synthesis and destroying the cell in one way or another. Any questions? Yes.
So the last one, the erythromycin, there is a production of like the protein, but it's just not what it's supposed to be.
Yeah, exactly. And I'm not going to go into like the actual mechanism, but there is, the protein eventually gets destroyed because it's altered, and that kills the cell, but it will be trying to make amino acids in that peptide, but it's not. It's gonna. So this is what I'm gonna get into next. So basically, in translation, you're supposed to get a protein, but that's not always what happens. Sometimes those proteins are misfolded. Sometimes they don't look proper, and so the cell then has mechanisms to process the protein and see, okay, is this a functional folded protein that I can now use to make a functional enzyme, or do I need to modify it or degrade it or just get rid of it completely? So that's kind of one of those in-between steps. Yeah, that's a good question. Ultimately, antibiotics will destroy the cell regardless. That's what they do. But where they bind is kind of a little bit different. And in some cases, they can bind this P site and still make these wrong amino acids, or you can have or the wrong peptide bond formation between the amino acids, or you can just completely inhibit the peptide bond formation. Does that help? Yeah. Okay. So yeah, kind of going off of that, after we have proteins being synthesized, the translation isn't the last step in producing a functional molecule. So finally, we've made a polypeptide, but this isn't the end. And so the cell has to now make this polypeptide into a functional protein or an enzyme. And it can be modified in a number of ways. And so, like I said, if the protein is damaged, the cell also has mechanisms to remove it from the cell. And so some of these ways are, it can be modified, folded, and degraded. And so, yeah, just kind of a little bit of, intro here. A healthy cell cleans the house by degrading these damaged or unneeded proteins. So translation isn't the last step. You don't get the functional protein at the translation step. You're getting the polypeptide sequence that makes the protein, but then there's steps after to be able to say, okay, this is a good protein I want to keep and I want to make it functional, or it's damaged in one way or another and I gotta get rid of it. So protein processing after translation. So after translation, a lot of these proteins can be cleaved to either inactivate or activate it. So these proteolytic cleavages, I'm going to talk about that. So we talked about how that first amino acid, N-formyl methionine, which is the start codon, so that one is usually still attached. And a lot of times, a lot of proteins in bacteria, what they do is they have this N-formyl methionine cleaved off. This only happens in bacteria and not eukaryotes. This is important, and I'll tell you why. So basically, the reason why this is important and it only happens in bacteria is because we can take advantage of this in human health. So if you think about it from the perspective of having an infection, for instance. So say you have a bacteria in our bloodstream, and we have our immune cells that are trying to fight off that bacteria. Our immune cells can take advantage of the bacteria and the protein that's being translated having that N-formyl methionine because we don't produce it, so our immune cells are able to recognize it and know that that is part of the bacteria and we need to chase that down and not something else that our own cells make. So I'm going to show you a little video here of how this works. Okay, so here we have the neutrophil. Here's just our own red blood cell, and over here we have this diplococci, which I think is a Staph aureus, so it has 2 cocci. This is the neutrophil, and it's chasing it. And so all these other cells are our own red blood cells, and so you can see that this neutrophil knows only to chase this bacteria and nothing else. And the reason is because these neutrophils are attracted to that N-formyl methionine that's made by bacteria. So the neutrophils are able to recognize this protein processing step after translation. And so, yeah, this is actually a really nice example of chemotaxis. So we talked about chemotaxis in the past, and it's either an attractant or a repellent that our cells can be moved towards or away from, respectively. And so N-formyl methionine being released from the bacteria is that attractant that is allowing the neutrophil to sense that there's this N-formyl methionine in the extracellular environment, and it continues to chase it. And so every scientist has to ask the question, well, how do you know that it's N-formyl methionine that is chasing and not something else? Well, we obviously do wet lab experiments to see this, and so here we know that it's N-formyl methionine because I'm going to show you here. This right here is a pipette tip, and so we had N-formyl methionine in this pipette tip, and by we, not me, some scientists out there, basically they put N-formyl methionine into the cell, and so this is also a neutrophil here, and so you can see what happens here.
In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source.
Yeah, so that's that. So does that make sense? So it's a way that our immune system can target things that are strictly made by bacteria and that are not made by our own cells. So we're talking right now about before we jump into folding the protein and doing anything else with it, we're processing it. And so the bacteria are processing it. And so one way is removal of that N-formyl methionine, but there are other ways to process proteins after translation. And you probably have heard of these post-translational modifications before, like phosphorylation, adding a phosphoryl group, adenylation, acetylation, lipidation, methylation, glycosylation. You don't have to know all the details of these, just know how, know what they are and know that these are protein post-translational modifications that can occur. So there are 7 total. The first one, where I've given you more of an in-depth example, is removing that N-formyl methionine, and then there are these different types, which basically just add or remove some, structure to the cell in order to modulate the activity of these proteins by either turning them on or off, depending on if the cell needs them. Any questions? So, so this is, that all was at still the primary sequence level. But now let's talk about folding that protein into a 3D structure. So we're going into the actual protein folding here. Some proteins can fold on their own, but many of them require some help by chaperone molecules, which are also proteins that just help fold other proteins. So they're chaperones, hence the name. We're going to talk about a couple of different kinds, and so just to give you guys some context of how these chaperones work. The first one is DNA-K. So this one is a chaperone molecule, and so basically it clamps down on the polypeptide to assist folding. So think of DNA-K as the palm of your hand, and so the unfolded polypeptide will land in the palm of that hand, and there are these electrostatic interactions and forces that help the chaperone protein to kind of cup the protein, and it's those electrostatic charges that help to fold it into the globule. So the DNA-K protein, just think of it as something like in the palm of your hand. That protein, the unfolded protein, kind of just sticks to it and it cups it and it's able to fold it and then release it. So it's more simple. The second one is this GRO-EL, GRO-ES complex. So basically these are two proteins, these two chaperone proteins that are, that come together to make this complex, and they together are able to fold proteins. And so I'm going to talk about how this works. It works in a very different way from the DNA-K protein. So here you have this really complicated barrel. So I'm going to call this like a barrel structure. And so this is the GRO-EL portion of it. And so here we have the GRO-ES protein that actually acts as a cap on top of the GRO-EL. And so in red we have the GRO-ES. The GRO-ES is the cap. And so it controls the entrance or the exit of the polypeptide sequence into this barrel chamber. So this is just a top view image of the cap, and this is the GRO-EL barrel chamber there. And so basically what happens here is, what you need to know is that within this barrel is where the protein is folded, but it has to get in here somewhere, and it's this GRO-ES that controls its access to the GRO-EL. So ATP is used by both. So here you'll see that ATP is involved here, and it's also used here, but it's not actually the ATP that's helping fold the protein. It's the electrostatics within the cell of the polypeptide sequence interacting with these chaperones. That's what folds the protein. But just know that in this case, ATP is controlling the entrance and the exit of the protein from the chaperone. And so I'll explain this a little bit more. And so here I have a little movie for you to show you this. So here we're zooming into a bacterial cell right there. And then we have this GRO-EL barrel. And so this GRO-EL barrel, and then you have this GRO-ES chaperone protein. And so this GRO-ES caps one side, but the top is open, so the unfolded protein can come in. ATP also comes along in yellow there, and it's necessary for the entrance of the polypeptide, and then that allows, then when the polypeptide enters the barrel, the GRO-ES protein can then cap the top of it. Just some notes from the research article where this came from. So GRO-ES is able to cap it, and then once it caps it and that protein is folded, you get the release of the folded protein, but then the bottom part was open, and so you saw another protein go in, and so that was just another unfolded protein. So first unfolded protein comes in from the top, there's a cap, ATP hydrolyzes, it folds the protein, it comes out. That bottom part's open, another unfolded protein can come in, and then it comes out from, the folded protein comes out from the top. You get ADP released because there's hydrolysis. So this is just the way it's going really quickly. Does that kind of help you visualize what's happening? Okay. So it's a very controlled fashion with the caps coming on and off. So next, we're going to talk a little bit about protein degradation, so cleaning the house, essentially. And so the cytoplasm has to constantly surveil for useless or degraded proteins that can hurt the cell and then have to be destroyed and recycled. And so these degrons are these proteins that contain degradation signals. So the degradation signal is the protein with a degron, so a degradation signal on a protein. And so these degrons dictate the stability of the proteins. And so what I mean by that is there's usually this N-terminal rule where on the N-terminal side of the amino acid of the protein, that whatever that amino acid is, it correlates directly with the stability of the protein. And so you don't really have to know much detail about this, but just to give you an example to help get this this point through, this point across is some proteins have like leucine, and leucine usually makes it very unstable, and they have a really short half-life that's maybe about 2 minutes. And so that's having that leucine there is a degron, an example of a degron signal, because it's impacting the stability of the protein. But yeah, so it's these degradation signals that allow it to know, do I need to remove it or not based on its stability. And so misfolded proteins are recognized by proteases. So proteases are proteins that can destroy or break down proteins. And in part, one way that they do this is by the hydrophobic regions being exposed. So the hydrophobic core is supposed to be on the inside, just like the fatty acid, the phospholipid bilayer, where we have the hydrophobic inner core and we have the phospholipid heads that are hydrophilic on the outside. Proteins are similar, and this is just the way life works. When something's hydrophobic, it needs to not be exposed to the extracellular environment. And so basically, if there is a misfolded protein that has the hydrophobic region being accessible to the outside, it's going to be recognized by a protease, and it's going to then be used. The protease is going to chop it up and into the amino acids and then recycle it to be reused by this ribosome. So bacteria contain these ATP-dependent endoproteases. Basically, just know that these are proteases. You don't really need to know these names. They're just for context. If you're so inclined, you can read about it in the chapter portion of the book. But these proteasomes are protein-degrading machines, and these are also found in eukaryotes and in archaea as well. But I do want to kind of talk a little bit about how these proteasomes or proteases work. Any questions on that so far? So this protein degradation machine, or the proteasome, is very similar to the manner that we just talked about in the GROW-EL and ES system, where you have this inner chamber and then you have these two caps, the CLPY, on the outsides. The active site here, note, is in the middle, similar as the GROW-EL ES system. And so this chamber has these caps that modulate whether the protein can get into that chamber or not. But this time, instead of this chamber just helping the protein fold property, there are proteases in here that basically have enzymatic activity and help the protease chop up the protein to not make it into the barrel. So in here, there's scissors instead of protein folding machinery. So these are structurally similar to eukaryotic and archaeal proteasomes. This is just a comparison of very similar in size. In archaea, where we have 12 nanometers by 15, we're at 17 by 12.5 here, and you have similar sections. So we're going to go through just one example of how E. coli protein folding works versus degradation triage pathways. So when the protein finally exits the ribosome, the cell has a choice. Do I fold it or do I destroy it? And so This is essential for bacterial survival, especially during changes in the environment or stressful situations, right? So they are always in this fight or flight mode. And so here, I'm just gonna go through this. So you have this DNA K protein, chaperone protein. DNA J is just very similar, don't have to know that, but remember DNA K is a one way to fold the protein, in addition to the GRO-ELES complex, but DNA K here usually will bind to the nascent protein, and so this is the mRNA, and that's being translated, and so it can either, bring in the molecule here and fold it properly, and so that would lead us to step 2, where the protein is folded directly into its native protein form, where it's active. But the cell actually has to make a choice. So if this protein isn't going to get folded because there's something wrong with it, it's got to go through a degradation pathway instead. And so here, usually, it will go to, if it's not properly folded, the protein can then be fed to the GRO-EL for refolding. So maybe it'll have to go through the more complex system of protein folding. Or if it's still not properly folded, then it may be passed to a protease for degradation. So it can go through one and then it's still not properly folded, and then the cell knows to just take it through another mechanism to hold it, or it could just go through degradation. So it's not really one way or the highway. The cell is smart and it knows, it has all these mechanisms in place to help fold the protein or degrade it. So this is called this fold or destroy triage system, and this is essential for the microbe to survive, like I said. Okay, so the last thing I want to talk about is there are some proteins that are destined for the membrane. Remember, we talked about membrane transport proteins, or like substrate binding proteins and porins, just proteins that have to be embedded within the cell membrane. So far everything we've talked about is, up to folding the protein or degrading it, and usually those ones will just be out in the cytoplasm, but there are some that are destined for the membrane, and so those have to go through another sort of traffic control in order to be embedded in the right place. So they require these special export systems. And so these proteins that are meant for the cell membrane are usually tagged with this hydrophobic N-terminal sequence of about 15 to 30 amino acids. And so what's important that you should know here is, what should be actually bolded, is that they're hydrophobic, and they're at the terminal or the end sequence. So these are hydrophobic, and I'll tell you why that's important in a second. But basically, these sequences tether the protein to the membrane and allow these proteins to kind of melt into the fabric of the membrane and associate it with that hydrophobic fatty acid tails of the phospholipid bilayer. So basically, there's this co-translational insertion mechanism that I'm going to briefly talk about, but what is important here is that you have these proteins and they have these tags that have these tags are called these signal recognition particles, or SRPs, and so those are hydrophobic. And for proteins that are going to be membrane bound, usually they're bound in the fatty acid layer of the phospholipid bilayer, because those are also hydrophobic. So here we have a protein that's destined for the membrane, and so what happens is the SRP, this signal recognition particle here, will bind to the signal sequence, and it basically tells the ribosome to stop translation here, because the ribosome is trying to actually make the entire functional protein, and so this SRP, the signal recognition particle, really paralyzes the ribosome, and so it doesn't resume translating the protein until it encounters this FTSY protein. And so basically, once the SRP signals to the ribosome to halt, pauses translation, it's bringing it to this system here, the SEC-YEG. And so you don't have to really know all of these words. You do have to know what an SRP is and what it does to the ribosome. And so here, in theory though, I'm gonna go through what these are just for your context, but the paralyzed ribosome is then brought to the membrane, and it specifically interacts with this FTSY, this membrane, and so it becomes this membrane-associated protein that can bind and deliver the complex to this channel of SEC-EY, or SEC-YEG. And then here you have this GTP hydrolysis, where I mentioned earlier that there's a lot of GTP hydrolysis that helps these mechanisms, go and function. And so this GTP hydrolysis is essentially what transfers this peptide ribosome complex to the channel, and then the signal sequence is released and it is able to embed it into the membrane. So these are the three steps that you should know. So basically, like I said, ribosome is paralyzed by the SRP, so until it encounters this FTSY, and then translation can recommence, and the translated peptide is fed into this SEC-YEG translocon, or another translational protein. That's why it's called co-translational export. And then the transmembrane sections of the peptide are then released into the membrane by a lateral gate, and so those hydrophobic regions are able to then kind of mesh in and blend into the hydrophobic region of the membrane. And so the reason that this is important as a way to embed a protein into the membrane while it's being translated already, you have to halt that translation until it makes it to the FTSY in order to resume translation, is in order for the protein to get embedded into the membrane, there has to be those hydrophobic amino acids. That presents a problem for proteins that are in the cytoplasm, because I just told you a few slides ago that there are proteases everywhere that are trying to chop up proteins that have hydrophobic regions that are exposed to the outside. So this is one of the reasons why we have this co-translational export mechanism. We have to halt translation, and we have another co-transporter that it has to meet before it can resume translation, because otherwise there are these proteases that are gonna just chop it up and release it. So yeah, those proteases are in the cytoplasm here. And so it needs to be halted, because otherwise those proteases will chop it up and degrade it. But we need those hydrophobic regions to be able to be embedded into the hydrophobic regions of the membrane. And so that's why we have to stop translation. Does that make sense? Okay. So this was a continuation of chapter 8. I know it's heavy and there's a lot of different mechanisms, and although it might make sense as I go through it now or the first time, once you're putting it all together and trying to remember all 3 steps of like translation, transcription, the ribosome and the tRNA and all of that, it could get confusing. So I have a worksheet 3 for you guys that goes through the last 3 lectures, lecture 6, which was on viruses and phage, 7 on transcription translation, and then today's lecture 8, protein processing. So these will help you guys, review and test your knowledge on everything that I've talked about so far. And so we'll definitely go through all of these and have like a review. Half of next, or on Wednesday, we'll have like a review session on all of this. And then we will start the next lecture, and then we'll have 2 more lectures after this before your exam, which is going to be after your spring break. So just to kind of give you guys a pause and a little review, pick one of these up and complete it by Wednesday, and then we can go through them and bring your questions as well on Wednesday so that we can talk about everything and answer your questions.
I have a question about so when the translation is stopped, does that just mean the proteases that are chopping up that hydrophobic region aren't produced?
They are, they're just floating around in the cytoplasm, but they don't recognize it because that SRP molecule binds it and brings it to that co-transport area.
So they're there, but they don't recognize it? They
don't, yeah, because now we have that SRP molecule that's binding it.
Oh, carrying it.
Carrying it over, yeah. So it's removing all of that protein from the cytoplasm and taking it to that co-transport area.
All right.
I just have to go back to that. You're welcome.
See you tomorrow, lab.
Yeah, let me write this down and then. Love's at 4. I think it's at 3:30. Then I'm going to that at 4. After